Good morning, everyone. People sitting on the back, I request to please come forward. I extend my warm welcome to all of you. Today, we have gathered here to celebrate Festival of Science and Technology. National Technology Day is celebrated annually on May 11 in honor of India's scientists, engineers, and their accomplishments. So, we all are here today to recognize and celebrate ourselves. Let's take this opportunity to compliment ourselves for our hard work and achievements. This day commemorates the successful Pokhran nuclear detonation test in May 1998. Apart from Technology Day celebration, we are also going to release CSIR-CDRI annual report of financial year 22 to 23, which makes this gathering much more special and meaningful. So to formally begin the function, with all due respect, I invite our guest, Dr. Manjari Bakre, founder and CEO of OncoStem Diagnostics Private Limited, Dr. Dhananjay Dendukuri, CEO and co-founder of Akira Labs, our director, Dr. Radha Rangarajan, organizers of this event, Dr. Atul Goyal and Dr. Ashish Arora, to please come up on stage. It's an honor to have distinguished dignitaries accompanying us today. I request Director Ma'am to welcome our guests with flower bouquet. May I please request our Director Ma'am to deliver welcome address?
Distinguished guests on the dais, colleagues, friends, and my dear students. Welcome to all of you to celebrate National Technology Day 2023. It is actually celebrated on the 11th of May, as Bhavana said, but we have decided to extend the celebration and do it on a slightly more convenient day as per calendars. So an extended National Technology Day celebration. All of you know this, that we commemorate the significant achievements and contributions made by Indian scientists, engineers, and technologies towards the growth and development of India. This is the 25th year of celebration. On the 11th of May 1998, when India conducted the Pokhran 2 nuclear tests, the then Prime Minister, Shri Atal Bihari Vajpayee, was so moved by the enormity of the achievement that he decided to dedicate the day to scientists and technologists. And as members of a CSIR laboratory where research and development are core to our existence, the celebration is also a validation of our purpose and commitment. While preparing for this event, I found out that 11th of May has special significance for CDRI. On this day in 1999, actually the first National Technology Day celebrations, Prime Minister Vajpayee launched Alpha Beta R Theta, an anti-malarial drug with the brand name EMAL. So this drug, as you know, was discovered and developed by CDRI with support from CMAP and is currently marketed in several countries. And as per today's data, the product is now available under 521 different brand names and has saved countless lives. Indeed, if one is to reflect on the advances of science and technology in India, it is fair to say that we have come a long way. A scientific and technology-intensive approach to development was part of the vision of the founding leaders of India. Over time, this has paid off, whether we look at space, biotech, pharma, IT, or agriculture. there have been major contributions on the scientific and technological front. This has led to job creation, value creation, and ultimately the growth of the economy itself. In the future, too, solving some of our more pressing problems related to climate change, waste management, drugs, vaccines, and even urban infrastructure will have to come from sustained efforts in scientific research and development. Of course, India's current R&D spend, currently at 0.6% of GDP, will have to increase significantly, both in the public and in the private sphere, to tackle the hard problems that lie ahead of us. This year, the Prime Minister addressed the nation on the 11th of May, in which he said that creating an inclusive ecosystem for growth, innovation, and sustainable development goals requires the use of technology at every step. In line with this, the theme of the technology showcase at Pragati Maidan was titled School to Startups, emphasizing the role of youth, and more particularly startups, in shaping India's future. At CDRI, we have a history of working with startups and MSMEs, and I hope to expand and deepen these engagements. So today, I'm delighted to welcome two trailblazing entrepreneurs and courageous thinkers, Dr. Manjari Bakre and Dr. Dhananjay Dendukuri. Both are pioneers and started companies in the early days of the startup ecosystem. Both trained as scientists and were passionate about research-driven product development. Their companies aim to improve patient care through diagnostics. They have forged their way through the many ups and downs of running companies, persevered, and are role models for innovators and entrepreneurs. Dr. Manjari Bakre got her PhD at IISc, then worked at Mount Sinai School of Medicine and at UCSD. Then she worked at Avasthagen and then Philips Research Center before starting OncoStem Diagnostics. The company has developed innovative prognostic tests that assess the aggressiveness of tumors, and their flagship product, Can Assist, uses a proprietary algorithm to identify a patient's risk of recurrence of breast cancer, which then guides therapy. So no doubt this has enormous impact on cost and quality of life. The company has had marquee investors such as Sequoia Capital and Artiman Ventures, and has a partnership with Zydus for sales and marketing. Achira Labs is a medical diagnostics company that manufactures a microfluidics-based immunodiagnostic system for measuring hormones, detecting infectious diseases markers, as well as testing semen quality for IVF. The company also offers design services to those building microfluidic systems. This is in fact how I got to know Dhananjay in the first place. Cipla acquired a significant stake in the company in 2016. And Dr. Dhananjay Dendukuri studied at IIT Madras, then got his PhD at MIT. He worked at Connexios before founding Achira. Manjari, Dhananjay, thank you so much for accepting our invitation to be with us today, and look forward to your talks. I want to also share some good news. And the 11th of May seems to be an auspicious day for CDRI. So on this 11th of May, Cipla filed a clinical trial application for L-ormeloxifene, the L isoform of centromin. Dr. Kamboj is here in the audience. I'm glad to see you here. So this is our best known drug, once weekly oral contraceptive, and it was developed from bench all the way through clinical development at CDRI. And this particular milestone is a very major one for all of us. I want to congratulate the entire team, Arun Trivedi, Ravi Bhatta, Rajesh Jha, Naseem Siddiqui, Sharad Sharma, and Vivek Bhosle, and their students and technical officers who worked together on this project. A round of applause for the team. Another milestone was that a RT-PCR kit for COVID-19 that uses indigenous probes, a novel universal quencher, and dyes that were innovated in-house was validated by the National Institute of Biologicals and found to be 100% sensitive and 98.8% specific. With this data in hand, the industry partner, Biotech Desk Private Limited, can take the next steps towards marketing authorization. I want to congratulate Dr. Atul Goel, Dr. Ashish Arora, Dr. Neeti Kumar, and their students, and Naseem Siddiqui for the sustained efforts that have brought us so far. Thank you very much. Going back to today's program, as Bhavna mentioned, we will release the institution's annual report for the year 2022-23. This would not have been possible without the contributions of a large number of people. I want to thank the editorial team headed by Anand Kulkarni, area coordinators, heads of division, and all contributors for the many months of work that went into the preparation of this document. I also thank the organizing committee of today's program, headed by Dr. Atul Goel and Dr. Ashish Arora, for their efforts in making this program happen. Thank you. Before I end, I want to leave you with Carl Sagan's quote: "Somewhere, something incredible is waiting to be known." So let us stay dedicated to the incredible endeavor of scientific discovery. Thank you.
Thank you, ma'am. Now it's time for the memorable moment, unveiling of CSIR-CDRI Annual Report 22-23. It contains comprehensive report of CDRI's activities and achievements, summary of past financial year, along with our current and future research focus. Thank you all. May I please request Dr. Atul Goel to introduce Dr. Manjari Bakre to the assemblage.
A very good morning to all of you. It gives me immense pleasure to introduce today's speaker, Dr. Manjari Bakre, the founder and CEO of OncoStem Diagnostic Private Limited, Bangalore. Dr. Bakre, a cell biology and cell signaling veteran, has 30 years of experience in research and academia. She received PhD in cell biology from Indian Institute of Science, Bangalore. Post PhD, she was worked in prestigious institutions of US and Singapore in the area of cancer biology. She moved back to India in 2008 and led a group in cancer drug discovery at Avesthagen, followed by multidisciplinary research on point of care diagnostics at Philip Research in Bangalore and the Netherlands. She founded OncoStem Diagnostic Private Limited in 2011 with the vision of developing and delivering innovative, cost-effective and reliable tests for personalized cancer treatment and planning. Her Cancer Assist Breast flagship product for breast cancer patient is being sold in India and internationally for the last 6 years. Dr. Manjari is a recipient of multiple awards like Young Scientist Award given by International Union of Biochemists and Molecular Biologists in 2006, Best Entrepreneur Award, Global Women in STEM Startup in 2017, Entrepreneur of the Year Award from Indian Achievers Forum in 2021. She has published many peer-reviewed journals such as Nature Medicine and has multiple patents in her credit and mentored several PhD students. I'm happy to share that OncoStem has won NASSCOM Best Innovation Award, received Aegis Graham Bell Award in 2021, given by Ministry of Electronics and Information Technology, Government of India, Today she is here with us to deliver National Technology Day Award lecture entitled Does Every Breast Cancer Patient Need Chemotherapy? Role of Made in India Innovative Test in Decision Making. With these words, I invite Dr. Bakri to deliver her talk. Dr. Bakri.
I think I So first of all, thank you, Dr. Radha and the team, Dr. Atul, Dr. Ashish, for having us here, team of CDRI. It's always a pleasure speaking to colleagues from academia and speaking to aspiring students here who are here to really motivate you further to do something really out of your comfort zone. So always focus on doing something out of your comfort zone, because that's the only way to progress yourself, your careers, and the country. So let me start. I think Dr. Atul has given the introduction, so I won't go any more details on that. So the title of my talk is, Does every breast cancer patient need chemotherapy, and are there any prognostic tests to assess the same? So cancer, what does cancer, the moment you say cancer to anybody, the only image that you get in your mind is chemotherapy and patients losing hair and so on and so forth. So the central question is, does every cancer patient need chemotherapy? And the short answer is, actually, every cancer patient does not need chemotherapy. And so then let's go back to the why. So why is chemotherapy given? So broadly, cancers are liquid tumors, the blood-borne blood cancers, and the solid tumors like the organs, liver, lung, bone, breast, brain, etc. So today I'm going to focus on solid tumors, and in that, breast cancer. So on the whole, the treatments for cancer, especially in solid tumors, are surgery and followed by radiation. So surgery plus radiation are considered local treatments. So locally, they would remove the tumor from the breast and radiate that area to remove to kill any cancerous cells that may be there. So that's called local treatment. As against systemic treatment, which means treatment given through your IV, through the blood, which the drugs go anywhere. And that can be chemotherapy or targeted drugs like Herceptin, antibody drugs, biosimilars or ADCs, or of course tablets like anti-hormone therapy that is given in breast cancer for hormone positive breast cancer. So those are targeted therapies, and so but they fall under, of course, systemic therapy. So chemotherapy is a systemic therapy, and when you look at, I'm talking about cancers which are diagnosed very early. So every breast cancer patient who is diagnosed early with small tumors, 1 centimeter tumor, 2 centimeter tumor, up to 5 centimeter, they're easily resectable. Surgeons can go ahead and operate it. And despite clear margins, that is the tumor that is removed when it is checked in pathology lab, on the edges of the margin, there is no cancer, which means the cancer has been removed completely. Even then, patients can get radiation to ensure that no growing cell is left behind. And after that, there is a hormone therapy for every hormone positive breast cancer for 5 years in the past. And importance of why hormone therapy is given is to prevent the hormone. This is hormone responsive cancer. So the anti-hormones prevent the cancer from growing. And this is why hormone therapy is extremely effective. It's a part of systemic therapy, and you can never rule out usefulness of hormone therapy along with chemotherapy when the cancer doesn't come back. So why is chemotherapy given? To prevent cancer recurrence. And when cancers are small, like in breast cancer, which is not a very aggressive form of cancer like pancreatic, etc., and you have clear margins, the surgery is done very well, the tumor is removed entirely, radiation has been given, patient does have the option of taking hormone positive, because she is hormone positive, hormone therapy. In that background, the benefit of chemotherapy is, depending on whom you talk to, surgeons versus medical oncologists, it can range up to maximum of 15%. But how do you find these 15% patients, 15 out of 100, who we should get chemotherapy? Nobody knows that today. So today, typically 95% patients in India get chemotherapy, and that's the one size fits all therapy that is absolutely not required for every patient. Patient who is 66-year-old who has 0.5 centimeter versus somebody who is 35 years old with 2 centimeter, both of them, there is no need to get chemotherapy. In fact, even two patients of 45 years of age, both of them have 1 centimeter tumor, lymph node negative. One may need and one does not, but how do you know that which one of them needs? And that's the part that's where prognostic tests help. So the problem really what I had seen in India is there are, of course, the breast cancer market is tremendous across the globe, including India. In India, we get, as per some census some time back, is 1.8 lakh new breast cancer patients per year. This number hasn't gone up significantly since 11 years of my work. So my suspicion is highly undercounted number. And but irrespective, 1.8 lakh new breast cancer patients per year, and out of that, we are looking at almost 50% patients who are eligible. That is, they have small tumors and they are hormone positive cancers. So all of them, so in India itself, we have about 65,000 patients, and worldwide there are 900,000 eligible patients. So out of these, 95% patients of early stage breast cancer get chemotherapy, but actually only 15 out of 100 really need it. And how do you find these 15 patients is by use of prognostic tests, because chemotherapy is extremely toxic, and it is not only physiologically but also financially in country like India, where primarily the expense is out of pocket. And prognostic tests have been helping patients in the West, in US and in Western Europe for a decade now to prevent excessive use of chemotherapy. However, those tests are they're just prohibitively expensive for our patient. The cost is $4,000. And in that is absolutely unaffordable. From India, we may be able to send them about 100 or 200 tests per annum. So Western prognostics are neither affordable. So what is the option for our patients? And that was really on my mind. And when I asked many doctors, if I develop a test tomorrow, which is a prognostic test, which is cost effective, will you use it? So then they were very keen to use something like this. And so just briefly, prognostic tests are tests which really assess the chance of breast cancer recurrence or any cancer recurrence. And Oncotype DX and MammaPrint are the leaders in this field in breast cancer from US and Europe. They are in every major treatment guideline, and they have led to reduction in use of chemotherapy, and thousands of patients have been benefited due to them by not taking chemotherapy, and thereby reducing the toxicities and improving quality of life. And when are they done? So this is the one question I often get, how many times one should do prognostic tests? You should do it only once. When is it done? So patient diagnosed with hormone positive, HER2 negative breast cancer, which is the best form of breast cancer to treat. And early breast cancer, post-surgery is when prognostic test is done. While the patient is recovering from surgery, it is the ideal time to send the tissue to the lab to do it. And while she is recovering, we can assess the tumor and say whether the patient has low risk of breast cancer recurrence or high. And typically it is 70-30. And the 30% patients only should take chemotherapy and sparing the remaining 70%. So it is done only once on the tumor tissue that is resected during surgery. In some cases, we can do one on a biopsy sample too, but the biopsy sample has to be bigger than on those fine needle samples. So the Western prognostic tests have been lately, more and more data is coming out that they are not ideal for Indian patients, or they are not ideal for many patients, including African Americans, Ashkenazi Jews, etc., even Asians, like there's Japanese and Koreans as well. So why is that difference? So that is because the population-wise Asian breast cancer patients are diagnosed. Asia is a younger, people are overall younger than the West. And the disease diagnosis in breast cancer is at premenopausal age. At around 48 is the median age, as against 60 plus in the West, around 55 to 60. And higher tumor burden. Our tumors, for variety of reasons, awareness, denial, just assuming that things will go, if somebody ignores a lump in the breast, it will disappear, etc., etc. Our patients come forth when they are having the pain that is unbearable. And so it is higher tumor, the tumor burden is slightly bigger, or maybe one lymph node is involved, etc. As against small, and mammography is not a rampant screening solution in India. And so that's another reason why, unlike in the West, our tumors are 2 centimeters, where in the West, because of mammography, it's 0.1 centimeter, 0.2 centimeter tumor. So, and that due to all of this, the biology of the tumor is different at diagnosis. So our patients are younger, and the biology is mainly node positive disease, grade 3 disease, tumor grade, and high Ki67. For what it's worth, it's a marker of proliferation. So we needed a test which will encompass tumor biology of our patients. And that I felt was a need of the hour, because oncotype DX has been shown to work differently even in African Americans, let alone Asians, because they are not well represented in the development or in the validation sets. Mammaprint has been shown to work diagonally opposite in Japan and Korea versus how it works in the Europe. So OncoStem was founded in 2011 with a mission to develop innovative prognostic tests to personalize women's cancer treatment and to improve the quality of life and reduce cost of care, with, of course, having better clinical outcomes. And today we are I'm happy to say that we have launched our first test 6 years ago, Can Assist Breast. It is only meant for hormone positive, HER2 negative early breast cancer patients. This is first of its kind, made in India prognostic test, and it's the first test in Asia. The only competition we have is Oncotype DX, Mammaprint, and Prosigna from the US. So what is Can Assist Breast? So as I mentioned, it is a prognostic test. It assesses the risk of breast cancer, distant recurrence, distant as in the organ wise. We don't predict local recurrence in the same breast. We predict recurrence in liver, lung, bone, brain in 5 years up to 10 years from diagnosis. Recently, we validated for 10 years predictions as well. And how does it do it? So we take patient's tumor tissue that is removed during surgery, and we do immunohistochemistry for 5 biomarkers, and that input, those slides are graded by trained oncopathologists, and then that information goes into our machine learning based algorithm, which is again proprietary algorithm developed by our statisticians, and then that gives us a risk score, and that risk score is a number between 0 and 100, and based on the risk score, we stratify the patient as either low risk or high risk for breast cancer recurrence. As I mentioned, it is only meant for hormone positive, HER2 negative early stage breast cancer patients. So this is another thing to explain the same. IHC for 5 markers, that information along with patient's tumor size, tumor grade, and the lymph node status, this information goes into the algorithm, we get a number. If this risk score is below 15.5, we call the patient low risk for breast cancer recurrence, and patients from 15.6 risk score up to 100 are considered to have a higher risk of breast cancer recurrence. So this validation so one thing, because you have a TNBC test in, so you need to develop algorithm on a set of breast cancer patients or whichever cancer patients samples and validate it on additional, totally unrelated patient samples. And the more validation, it's a numbers game. The more you do, the more power you have, the more confidence anybody has in your test. So you just have to validate practically till you are saturated in the market. So we still continue to do validation studies. And at the same time, as I mentioned just some time back, patent. Do not publish before you patent. I know there's always urge, I'll present in small conference, I'll present. No, don't. Just really focus on patenting first, and only then you publish it. So we have patented it in multiple countries. Patent is granted, and we have several scientific peer-reviewed things. And more importantly, accuracy is 95%, which is at par with Oncotype or Mammaprint. We have validated Can Assist Breast in multiple studies across the globe in 21 different clinical studies, 4,000 patients. And this really is the hardest part of my work. I mean, first hardest is, of course, raising funds from investors. And the second hardest is this. Sometimes when I look back, I think raising funds was far easier than this. So it is extraordinarily hard to work with clinicians because they are really busy. They don't get any single day for the week to do any clinical work. And they are inundated with patients, and it's just that they really don't have the time. They want to do it, but they don't have the time. And then plus all the bureaucracies and the number of approvals you need to work with any clinician from different departments within the hospital. So somewhere it fails. And so you just have to spread your net very, very wide and approach many doctors, many hospitals, and then finally, you work with 20 after approaching maybe 200. So and do this simultaneously. Time is your biggest enemy. So do it as simultaneously as you can. And that's what we did. And we worked also internationally in Germany, Spain, Italy, Austria, and the Netherlands. That's not listed here. So any lab developed test, one needs to show analytical validation and then validation in clinical format, and we have done all of that. And all this data have been published. And as a startup, guys, let me tell you, publishing is even harder. So when I was a PhD student at IISc, publishing was not easy from India at that time. And people used to journalists used to just return the paper saying, oh, this is all published. And we used to just argue back saying, what are you saying? This is not published. Show me one publication which shows this. And then they would say, yeah, you're right. Please send it back. We'll review it. We say, we already sent it somewhere. It is accepted. So that thing hasn't changed after 30 years. And because now I'm facing additional challenge of being a startup, which nobody knows. So first of all, journalists don't look very respectfully at startups and at industry on the whole, and least of all startups. And then an unknown startup from India doing this kind of a very highly competitive work. It was very hard to do our first paper and second paper and third paper. And then now it's getting better and better, but still it's not easy. I don't know whether it will ever get easy. So coming to the data, I'm not going to show too many data slides and don't bore you, but just want to convey the concept. What is prognostication? So this is what CAN-assist breast does. So if you are given a set of 3,000 patient tumor tissues and say, okay, tell me how many of these will recur versus not, we will run CAN-assist breast on them, and then we'll segregate. This is how the CAN-assist breast, this is the typical Kaplan-Meier curve. Y-axis is distant metastasis-free survival, and x-axis is time in months for 5 years. So CAN-assist breast will give you risk scores in such a manner that they are separated using KM curve. Patients are separated 70-30, low risk, and they have a higher distant metastasis-free recurrence of 94% versus high risk at 82%. All of these patients received chemotherapy plus endocrine therapy. So one can say there was some advantage of chemotherapy in these low-risk patients. So we also looked at patients who none of them received chemotherapy for whatever reasons we don't know. These are all retrospective studies. And even in endocrine therapy treated patients, CAN-assist is able to find 20% patients who are at low risk for breast cancer, who have high risk of breast cancer recurrence. So why is that? So this is the really important point, that currently clinicians are dividing patients and deciding on whom to give chemo versus not based on size of the tumor, grade of the tumor, level of Ki-67, age of the patient. ER/PR HER2, of course, we are talking about only hormone positive, HER2 negative. So clearly this shows that with this, they are going to go wrong 20%. They are missed out giving chemotherapy to these 20% patients. So CAN-assist breast not only helps you to prevent chemotherapy, excessive chemotherapy to patients who don't need it, but it also tells you maybe you have missed out because she has a 1 centimeter tumor and she's 70 years old. You think she may not recur, but that's not true. If you look at the underlying biology, the biology, irrespective of age, it's the tumor biology that will lead to cancer recurrence, not the age of the patient. Age of the patient will play a role, but tumor biology certainly triumphs, or tumor biology triumphs any anatomical features. And so that's why, because CAN-assist breast looks at 5 biomarkers other than ER/PR, HER2, Ki-67, we were able to capture the tumor biology of aggressive cancers. And we've also showcased that why we keep saying the CAN-assist breast high-risk patients should take chemotherapy. So this is another graph to showcase that the data look differently here. We looked at all the patients who were stratified as low risk by CAN-assist breast versus high risk. So in the low risk patients, we segregated to see patients who received chemotherapy plus endocrine therapy versus who received only endocrine therapy. Both these curves are overlapping, and all of them have distant recurrence-free interval of very high 96%. What does this mean? When CAN-assist breast patient calls patient low risk, whether or not you add chemotherapy to her regime, it's of no use. So these patients will do equally well with just endocrine therapy. On the contrary, when CAN-assist breast is segregating patient as high risk and you omit chemotherapy, you treat her with endocrine therapy alone, she is going to recur. Those patients are at high risk. They need chemotherapy. They would benefit by adding chemotherapy, and this difference in their survival of 90 to 89, this difference is what's called chemotherapy benefit. That by taking chemotherapy, these patients have improved life from these patients. So this is chemotherapy benefit, which is only seen in high-risk patients. Then, of course, we had to validate it globally to showcase that it works equally well. So this is the Indian versus Caucasian subcohort. Across all Caucasian non-Indian patients are clubbed together, where the patients were treated with chemoendocrine or endocrine. Can assist breast-based segregation is very wide, statistically significant, and the proportions are also similar. And all of this data has been published in different this thing. And the other annoying issue is hormone positives is they recur up to 15 years. So there were a lot of historical, I don't need to bore you with the clinical part of it, why they recur till late. So there were reasons why the way we were treated traditionally, they were given hormone therapy for 5 years. So there used to be another peak at 7th year, soon after you stop the hormone therapy. So that secondary peak that you see of recurrence is not because of first chemotherapy that you give in year 1. But doctors would still ask me, so can you predict 7th year recurrence? So we said, fine, we will do another 10-year validations, 10-year predictions. But those 10-year predictions are impossible to do with the retrospective studies by working with hospital directly. Doing 5-year follow-up, I had literally, I took every ounce of energy in me to do 5-year follow-ups, because patients don't come back to the same hospitals don't keep the files, and this and that and that. And so how do you follow the patients up? That is a fundamental problem. So 10 years is, forget it, impossible. It is not possible to humanly in India to do that work. So we had to approach all these international bodies who conduct, and India does not conduct any prospective randomized trials which are done fairly and have clear mandates to store tissues and distribute it for public for the greater good for the people. So we don't have samples. And so I had to approach every big agency in the world who have done prospective randomized study and explain that, hey, this is what I have developed and I would like to validate it. Would you be interested? Now imagine again, I'm a nobody startup person from India. Why would they give me their precious tissues? But it takes immense amount of perseverance and patience to explain to them why you want to do this, because our patients in Asia in general can't afford $4,000 tests. So after all that matha fodi, finally got a team. So this is a team trial, was a prospective randomized trial conducted in the Europe in 2001 to 2012, where they randomized patients to tamoxifen versus exemestane. These are two endocrine therapy drugs, and followed them for 10 years. And they had those tumor tissue samples with them archived in very well format. This is another one big thing to learn from all these trials. We can't archive our own samples in our own hospital, let alone a prospective randomized trial where tissues are distributed across 9 different countries, and in each country they have archived well. So a Dutch team from Leiden agreed to work with us. Again, I didn't know them from Adam, but it's the perseverance. I went and visited them and they said, finally, yes, we'll give you our tumor tissues to validate your test. And that tumor tissue was the one that had 10-year follow-up. So I was really keen. Through COVID, I pushed this study. They pushed it really hard, and finally it is done. It is completed. And they were it was a double-blind study. So I was literally crossing my fingers, and we had done our task of predicting and sending back, and I said, now you have to match and tell us. And they took so long. Finally, I said, look, I'm really curious. My fingers are crossed for a long time. So please tell me the results, because they're hurting now. So then they said it works so well. And this is the completely unknown data sets, cohorts, and 10-year predictions, and the cohort is really tough. Such trials, when Oncotype does it, they do it on a cohort of 1 centimeter tumor, lymph node negative, postmenopausal women, so very low risk. Look at this cohort. This is a cohort, it's node positive, 67% node positives, more than 55% have more than 2 centimeter tumors, T2 tumors, and more than 28% had G3 tumors. And despite all these high risk features clinical, 78% did not get chemotherapy. And on that background, we have to predict. So this was a really hard task, and we were really curious. And this just published it a month ago in Breast Cancer Research for 10 years, first time. And so these are reverse KM curves. And here you can see the low risk patients from high risk patients, very well segregated, very strong statistically significant p-values in not only in total cohort, in lymph node negative cohort, as well as lymph node positive cohorts. And we've also showcased this. There were 2 arms to this study, and in both arms, as well as in total cohort, test has predicted significantly the risk of recurrences. And coming to now the commercial part. So this is how today when we get test for running. So we took 5 years to develop, validate, get all the accreditations. In 2016 mid, we started selling. There were many mistakes we did while did not do much marketing. Many clinicians didn't know about us and so on and so forth. And at that time, we didn't have so much cash to do so much marketing. So these are all the lessons that for you guys also, don't shy away from marketing. Marketing is essential part of whether your test is terrible or your test is the best, marketing is essential. So more so if the test is good. So this is how we, when we get the tumor tissues, we take sections and we do Canas's breast and we say that this patient has 5% risk of breast cancer recurrence. And then based on that, and that is low risk. So you can see the risk score on this scale, and this is the cutoff. So for this patient, the risk score was 8.8. Risk of recurrence is about this much. And based on this, the patient will not get chemotherapy. And this was really hard to publish, but I have explained this before. And these are all the publications. We have 2 more which are coming out, and we are also in Asian Geriatric Oncology guidelines. Just last week that they went online. So that is our first guideline inclusion. And so why Canas's breast is because it has multi-country validation. It is affordable innovation. We are at 4 to 5 times lower than any American test. In private sector, government will be even lower. We use cutting-edge AI/ML based algorithms to predict. That prediction algorithm also took us 2 years to mature and develop to really predict it. And quality-driven, of course, we have US CAP, NABL, and ISO 13485. And it's the fastest from getting sample. We always report in 8 days, but majority of the time in 6 days. And in India, we are selling across India, in Sri Lanka, Bangladesh, Turkey, UAE. Recently, we started in Saudi Arabia and also in Vietnam. So really want to build on that. And about typically, so far, we have done 3,000 tests from 2016 mid until now, more than 3,500 actually. And 75% are certified as low risk for breast cancer recurrence. And it's very heartwarming to see that many insurance companies are reimbursing cost of the test to the patient. So we did a whole lot of work to create awareness amongst insurance payers because they are our biggest customers apart from patients. Doctors are customers, but doctors are not going to pay for the test. So doctors are customers in terms of creating awareness to recommend the test, to prescribe the test to the patient. In the end, it's the patient who pays or the insurance company. So you need to create awareness to them also saying, you will save your cost of chemotherapy for 70% of the patient if you pay for this test. And so they need to get that math in their brain and act on it to reimburse the cost of this test to the patient, and which is seen in some insurance firms, but not all. And CGHS, ECHS have done it, but not on a mandate basis, just on a case by case. And I am trying to understand why that happens. So if you have any leads to get into government insurance agencies or make them at least listen to us, I would really appreciate that. And these are real cases. Now these are really, so we've been in the business for 6 years. So patients who took Canas's breast in first year of our sales have finished 5 years. So I was dying of curiosity to know how are these patients doing. So if I had my way, I would follow every patient myself. But sometimes the patient's numbers change and this and that. So we follow every patient as much as we have numbers, and sometimes the numbers don't work. So this is the patient. She was diagnosed with breast cancer at the age of 31 in 2016 in our first year of sales. She was recently married and wanted to have a baby, and then with this breast cancer, she cannot have baby until she finishes endocrine therapy. Because she is 31, she had a stage 2 disease, and she was Ki67 was very high. So she would get standard therapy, which you are looking at 1 year of chemo, 5 years of endocrine minimum. And so she's looking at a baby at 37 years old. And as you know, it's not easy to conceive a child after finishing hormone therapy. You need to wean her off the endocrine therapy for about a year or two, only then she will conceive. So she is looking at, I don't know what age, or her option is do fertility preservation before you start the therapy. So she was very keen to have a baby. So in the end, it's the patient, it's her wish. And so the doctor decided to do Canas's breast. He suggested do this test, and based on this test, we will plan your therapy. She was low risk for breast cancer recurrence as per Canas's breast. She skipped chemotherapy entirely. Doctor allowed her to have the baby first. Right after surgery and radiation, baby was conceived and delivered. And then she was put on endocrine therapy, and she is doing very well for last 5 years, and she's a happy mom of a 5-year-old. No recurrence, no chemotherapy. So this was really, it's very, very, very nice to see her progress. And we have many such patients who have finished 5 years, who are low risk by cancer, finished 5 years and are doing well. And so these are some of their testimonials. There was a lady who was a rickshaw driver's wife, and she said this was really the most economical test. There was another patient who got the cancer the second time in another second breast, and she said the first time I took a chemotherapy, that was more than a decade ago, and then she said I really suffered, and now I realized there is this test, and as per the cancer's breast, she is low risk and she has been prevented chemotherapy and doing well. So now coming just to final slide on journey of OncoStem. So what does it take? So starting a company, I'm sure Radha will tell you more on this. It is a hard task. Dhananjay will, I'm sure, will agree. So it is not for the weak-hearted, but if you have a PP, the public-private partnerships, it will make your life a lot more easier than, because investors are far and few in healthcare in India. They are all in IT sector, because we are really, India is in where US was in IT sector in 60s or 70s, so that's the mindset we have now. So getting funding on long-term projects where you develop something and you sell, that takes time. In breast cancer, I need to do 5-year follow-up, so I can't hurry that up. So there is no app in the world that can do that. So that's the patient the investors need, which they don't have. So that's the hard task, and it took me. So 2011, but I was fortunate in that I was in some unrelated event, and so there was a person in the audience who said, "Oh, we like your idea, we want to fund you." And I said, "Oh, sure, let's talk." And so after 6 months, they actually funded us. And that's how the funding journey started. And this is out in 2011. But once you have the money, you need to start work. So get the hospitals to work with you, get the tumor tissues, get the follow-up, then develop the model, and then validate the model, sign up different hospitals in the US, patent it. Then the CAP model was finalized. We got all the accreditations, and look at the patent, proof of concept patent file 2012 versus first publication 2018. So that's what it takes. That's the time limit you are looking at to really patent first and publish later. And of course, you don't probably need this much time, but and then year on year, we kept publishing in different big conferences, etc. And then we had the validation done to 2000, 4000. Then we tried different models of sales. We worked with different distributors. Each model that we worked with we tried to develop our own team, but nothing is perfect. So finally, late last year, we worked with exclusive sales and marketing partnership with Zydus Life Sciences, and it's been just a quarter, and we hope to do well with that this thing. And I wouldn't have done this alone without my team. So happy to take any questions. Yeah, I had one more slide if I have time. I just wanted to show you guys the mindset change. So this is a story I really like, because it really shows the mindset change is so important. So this is about running. So there was this challenge of completing doing 1 mile, that is 1.6 kilometer in under 4 minutes. So the most fittest athletes in the US and Europe have not crossed that, doing 1 mile in 4 minutes. And it's not like they were at 5 minutes or 10 minutes. They were at 4 minutes 6 seconds, 4 minutes 1 second, but they were not under 4 minutes. And nobody was able to overcome that challenge. And actually, Roger Bannister, who was a doctor himself, he's a neurologist in the UK, and he was told by fellow doctors, he was at 25 when he broke that record, and you try to do this under 4 minutes, you might die. And so he took that as a challenge. He trained his mind to finish it, because he was not far. He was 4 minutes 1 second was the previous challenge. And then people so often wonder whether it's a perfect temperature, track, wind, shoes, outfits, what will make that break the barrier. And so this 4 minutes was not a challenge of physically, it was more psychological challenge. Because nobody had broken the record, nobody was willing to try it or give it a real rigorous try. But he did it. And on the worst day, it was rainy, wet track, nothing was ideal, but he broke that record, and he did it on 6th May 1954 on a wet track in front of just 3,000 people. and he did it at this time. And post that, within a month, within 46 days or so, somebody else did it. Within a year, 5 more people did it. And by now, there are almost 1,700 people who have done it. So what it took was just this one person changing it, the mindset change immediately that, oh, it's possible. So have that mindset for, this is for all the students, that, you want to achieve something, make sure you can make those inhibitors, whether it's PARP, it's CDK4/6, you can make it, you get your mind to train to that, and I'm sure you will achieve it. And thank you all for listening. Do you want to take questions now or later? Okay. Yeah. So we develop lots of models for that. And we systematically, that's why it took us 2 years to develop these kind of models, is to really keep trying different methods, different models. So the goal is not to develop it, a model with 100 genes, because that will increase the cost of the test. So then at the same time, you can't do one gene, because that may be, touch and go. So you need to have the optimum number of genes, markers, proteins in your test, which will be cost effective, but at the same time has high accuracy. So we developed many models and then chose this.
Is there an advantage to increase the number to 6?
There wasn't at that time from the methods we did. And so we narrowed down to 5.
That's sufficient to cover the entire diversity.
Looks like that, yeah. At that point, you don't know, right? You cannot predict for the entire future. 21 genes from Oncotype, they came down from, genome-wide analysis. So at that time, that was the best model that could have given us the best accuracy. So we stuck to that.
So ML is actually a hidden approach. You really can't say whether you will get lucky with 5 markers or not.
Probably, yeah. Yeah. But you do your best, and that's why you train it very well so that it will succeed in future. After some validation. Yeah. You need to have confidence in your product, your patent, your markers, your signature. Then you patent it.
What is an optimum number of patients that you should
at least
twice
the number used in development.
Okay.
It's very difficult to convince clinicians. Most of the time they suggest to the patients to go for the complete removal. Which itself will reduce the chance of recurrence. And even convincing not to give chemo. In fact, my very close relative suffered. They took all the precautions.
Okay.
She was just 49 plus. And even this K was not very high. But they suggested, no, it's always better to get rid of the breast. And then it's always better to give the chemo little bit. But as you said, okay, after the test, how you convince the clinician, no, hold on, don't give the chemo immediately. At the same time, how you consider whether they are removing the complete breast or only the partial tumor.
So the surgery decision we don't interfere with. Surgery decision is often taken based on patient's choice, whether she wants mastectomy, entire breast removal, or mastectomy with breast reconstruction, or just lumpectomy, just remove the lump. Okay, so that is entirely surgery and patient, we don't interfere in that because we are not even in that picture in there. Then, but now because of Oncotype has put in millions of dollars in marketing, everybody is aware of Oncotype DX. Our clinicians, a small clinician in Vietnam, everybody is known. They literally have done excellent marketing. They created the need and provided the solution. So everybody is aware of this, so they do, some of the clinicians do offer that test, but then the cost, and so what they didn't know, the clinician, is our test. So we needed to create awareness about our product. So that was the uphill task to go in every college oncology center and talk about our test. So if they see the data that is developed, validated, published, then that's when they really start trusting in your product and then start using it. And then it's there, the communication with the patient. And some patients are really well read. They go to clinicians with NCCN guidelines and say, hey, I have this, I want to do this test. Sometimes the doctors tell them, nahi, you need chemotherapy. They said, no, but I want to do the test. So that also happens. So it works both ways. But what is important from our doctor's perspective is to gain confidence in made in India products is to showcase the validations worldwide, publications, accreditations, all of those quality, that it is done in a quality driven manner. In private sector, it's 65,000 rupees.
In that case, what are all your plans to tackle that?
So that's the area, nobody can. So Oncotype DX is also patients have record in low risk. So our goal, my goal, or Oncosim's or any company's goal is to minimize that error. So our current NPV is 95%, and same is Oncotypes, Mama Print, etc. So we try to do our best to predict that in the low risk nobody will recur, but we are already saying 5 out of 100 will recur. So there is that. So so what is important from the doctor's perspective is to explain to the patient very well that there is a 5% risk. And if the patient understands that, and then they can say the patient was explained and agreed to do can assist breast. If they write that, I think court of law can protect them. That's what the doctors tell us. Because I said, this is the question you should answer, because doctors should tell, I don't prescribe, right? So we understand their complaint, their issue. But we also need to give them, and this is not just for cancer breast. They give so many drugs, and then sometimes patients progress on those drugs. So they also have to tell that every drug doesn't work on every patient 100%. Herceptin failures are so often. Progression on, despite taking endocrine therapy, so many progress, right? So there is a risk, there is a failure concept. No test, no drug is 100% accurate, works on 100% of the patients. But so long as you are aware and you have explained that to the patient, I think that does take care. Right.
There is something like if you check in between time periods, and then you try to associate, like if it has, the product has developed within last 4 or 5 years, how we can talk about the 10 years, and then whether data will associate, like as you showed some of the data which you have done with some other conducted studies,
Right.
but unless until this is done with these particular prospective samples which you are analyzing at the moment,
Right.
the confidence, I don't know, like
Yeah, no, we've already finished 5 years, and that data is already, we showcase in every meeting that we go, that in 5 years this is our accuracy, what we predicted versus what we are getting. So it's very close. In fact, we are doing better than what we predicted. Yeah, but that is and we are only predicting 5 years today. We have showcased that it validates for 10 years, but the model is for 5 years. Because to do prediction for 10 years, we need to develop another model or tweak this model. So we are doing only 5 year, and technically 5 year predictions are the best, because first chemotherapy is not going to protect you for second peak at 8th year. That is because the endocrine therapy was stopped. So, and now they have realized that extension of endocrine is done for 8 to 10 years anyways. So the second peak might actually just disappear. And even tests like on Mama Print, who were predicted to do 10-year prediction, they are best at 5. They have published it themselves. So in all likelihood, 5 year is the best chance.
All breast cancers.
No, only the hormone positive, HER2 negative confirmation is done, only then.
Yeah, because then otherwise it cannot be. Is it a first type test? No, or second, third, because you will be doing first pathology and then hormone positive and HER negative. Only then the test can be done.
Yeah, so the hormone positive, HER2 negative is typically done by the hospital. The test is only prescribed to such patients who are hormone positive, HER2 negative, and only they come to us, those tumors. So we don't repeat hormone positives at all. I mean, repeating the test for ERPR.
I have this technical question to understand how IHC is converted into a cutoff value.
So IHC is graded quantitatively.
Quantity, okay.
And those values for each of the 5 biomarkers along with tumor size, grade, node goes into our algorithm, which gives us this number, the risk score. And if that risk score is a number between 0 and 100, if it is under 15.5, patient is a low risk of breast cancer recurrence.
Okay, thank you.
So it's about 3%. Yeah.
Ma'am, I have two questions. First is that the 5 biomarkers that you have selected, are they all oncogenes or have you also included tumor suppressors? And the second is that why are you just focusing on positive breast cancers when TNBC is a major challenge in India?
So TNBC are 30% of the patients.
Yeah, but
hormone positives are more numbers of patients. So that was one. Hormone positive disease is the least aggressive amongst breast cancers. And so they really don't need chemotherapy. That is the second important aspect. TNBC, everybody by default will get chemotherapy. So our first thing was to really find a subset of breast cancer patients who have a low risk disease and still getting excessive chemotherapy. So how do we prevent that first? And they are the bigger in numbers. So that's why we focused on those cancers. What was your other question?
Ma'am, my question was that are the 5 biomarkers, they are all oncogenes or have you also included the tumor suppressors?
No, we didn't include tumor suppressors. We included the 5 markers. They
were probably up-regulated genes.
Yes, they are. Yeah, and these are involved in cancer metastasis. So that was really the science behind it. Okay, thank you.
Thank you, ma'am. Very interactive session. Indeed, very inspiring work which can revolutionize cancer treatment. Now, may I request Dr. Ashish Arora to introduce Dr. Dhananjay Dendukuri to the audience.
So thank you, Dr. Bhavna. Good afternoon, everyone. I have the honor of introducing our next illustrious speaker. It's fine. Dr. Dhananjay Dendukuri, who comes to us from Achira Labs, Bengaluru, a company he co-founded in 2009 and of which he is currently the Chief Executive Officer. Dr. Dhananjay did his BTech in Chemical Engineering from IIT Madras in 1999 and obtained Masters of Applied Science degree in Chemical Engineering from University of Toronto, Canada in 2002, which was followed by a PhD in Chemical Engineering from MIT, USA in 2007. So throughout he has studied chemical engineering. Dr. Dhananjay began his professional career as a lead scientist in Connexius Life Sciences, Bengaluru, where he was from 2007 till 2009. Achira, the company co-founded by Dr. Dhananjay in 2009, is a pioneering Indian company translating cutting-edge technologies in microfluidics to industrial practices in medical devices and diagnostics. Dr. Dhananjay leads a research team that works in the area of microfluidics, biological assay development, and instrument development. Achira develops and manufactures multiple diagnostic and medical device products. Dr. Dhananjay has also led funding for the company, bringing in investments from blue chip firms such as Catamaran Investments and more recently from Cipla. Being brilliant, it is natural that Dr. Dhananjay has received awards at every stage of his career. At IIT Madras, he received the Institute Blues Award, which ranks among the highest possible recognitions conferred by the institute on students. His PhD work on microparticle synthesis is also widely recognized as pioneering in the field of colloid particle synthesis, and he received the Centuria Prize for Best Thesis in the area of micro nanotechnology at MIT. More recently, he has received the National Technology Award in 2016 for commercialization of new technologies. Last year, he became elected fellow of the Indian National Academy of Engineering. Dr. Dhananjay has 24 issued international patents, several peer-reviewed publications, and 7 national and international government-funded major projects. So we are really privileged to have him with us today, and I'm sure that it will be a delight to listen to him. I now invite Dr. Dhananjay Dentukuri to deliver his National Technology Day lecture. The topic of his talk is Rapid Advances in Point-of-Care Testing through Microfluidic Technologies in a Post-COVID World. कोविड उपरांत विश्व में सूक्ष्म तरलता प्रौद्योगिकी द्वारा पॉइंट ऑफ केयर परीक्षण के क्षेत्र में तीव्र प्रगतियां. डॉक्टर धनंजय,
Thank you. That was impressive that you quickly converted the title into Hindi. Okay, great. Thank you, Radha, and Dr. Arora and Dr. Goel for the excellent hospitality and inviting us here for this National Technology Day. Very happy to be here at CDRI, and just heard an amazing talk by Manjari and showcasing some of the excellent science as well as clinical research that she's doing. So even for me, that was a very inspiring story to hear. So now shifting gears maybe a little bit, we are much more of an engineering technology company. So what you will hear is maybe more engineering than science, but hopefully some of it is applicable in the world that you see as well. Second. Sure. Yeah, actually, I'll keep talking because I remember my slides. So, so just to, first of all, just start with a little bit about the company itself. Our mission really as a company is to build POC tools that can do faster, simpler, cheaper testing. Really try and take all sorts of complex testing to very simple workflows, make it as simple as, let's say, a home glucometer, but do that for even more complex tests, whether they're immunoassays or molecular assays. Yeah, so this is good. And in terms of what we've done, we've built a platform technology that I'll be talking about today, which can enable this sort of load and go, as we call it, automation with quantitative results. So that can be done in a near patient setting. We, in addition to building new technologies, we also manufacture diagnostic products, including COVID kits and things like that. So we have an integrated R&D and manufacturing facility in Bangalore with clean rooms and licenses from CDSCO, etc. And as mentioned, we've got investment support not just from CIPLA and Catamaran, but we've been generously funded by various government grants, especially from BIRAC and Grand Challenges Canada, both of whom have been big supporters of the company. So in terms of what we are doing today as existing products, we're focused on maybe I would say broadly 3 different areas. One is more regular testing kits. So we made over 4 million kits, COVID kits during the entire pandemic. And we continue to do that as well as make some sewage surveillance COVID testing kits. but I'm not going to really talk about that today. These are on open RT-PCR kind of platforms. In addition to that, we have built, of course, microfluidic technologies for immunoassays and increasingly also for molecular tests. And we have some products in the IVF space as well, some of which are marketed by larger companies like Bharat Serum. So first of all, just to give you a little bit of context to this whole POCT space, it's a word that has been thrown around quite a bit, and understanding where we are with POCT is important. I think COVID was very transformative for point of care testing. It's a term that has been around, like I said, maybe for 20 years. But I think it was sort of at a very low level building up. But COVID, as you all can imagine, has really transformed POCT testing because everyone got used to the idea of doing testing at home, which is what the whole idea of POCT is, to eventually move testing as close to the patient as possible. And of course, nearly everybody sitting here has probably done a rapid test sitting at home. So that has got hugely enabled in COVID. But then to do more and more of these kinds of tests, you need technologies, especially I would say sample preparation, integration technologies, and that's where microfluidics can play a big role, because you want the workflow to be very simple, right? And I think that's the biggest challenge most people face. If you don't have a scientific or a lab background, even simple steps can seem quite complex. And that's where engineering technologies, microfluidic technologies can help automate a lot of that. And the context, again, from the Indian point of view, is microfluidic technologies developed in the Western context are very expensive to use in India, right? I mean, just to give some sense of numbers, I think any cartridge plastic consumable will not be sold, let's say, for under $20 to $30. That's probably the lowest price point you will find in the US. And you know, at that price point, you can all calculate, you will do an entire health profile in India. We're all continuously getting spammed messages from the big labs, do health profile for 999 rupees and 600 rupees and so on. So it's, those price points just don't match. So you have to be able to do this obviously at a number that makes sense in India. For microfluidic tools, what they do are three major things. One is, of course, miniaturize. So miniaturize both sample volume, which is primarily means can you use capillary blood as opposed to venous blood, so you can enable home testing. The second is you want to integrate a lot of features. It could be simple things like mixing, it could be dilution, it could be, of course, separations. All of this could be really miniaturize and integrate it together, and of course, automate it as one package. And there's been big movements in microfluidics-based POCT in the last few years especially, but just looking at what's happened over the last 50 or 60 years, to give you all some context, actually some of the tests are quite old, like the glucose strips and the home pregnancy tests are almost more than 50 or 60 years old. So they've been around for quite a while right now. And then Abbott iStat, which is blood gas electrolyte analyzer, again, very popular in the ICU setting. That is also, I would say, very commercially successful POCT instrument. And then we had a whole set of things that came up during COVID, including one of the homegrown stories, which is Mall Biodiagnostics, that also did extremely well and was actually used by ICMR, many ICMR labs to run COVID tests. And then there were several others as well. And I think if you think about what's enabled this beyond COVID, we all understand there was a need for lots of testing. But beyond that, there are two or three things that have happened. One is, I think, the mass manufacturability of these plastic consumables with miniaturized features. I think that's really gotten enabled in a nice way just through advances in engineering, incremental advances in engineering over the last 10 to 15 years. Second one is low-cost electronics. I think, needless to say, you know, your smartphone today has cameras which are probably better than many scientific cameras of 20 years ago, you know, and those are all maybe $10, $15, $20. So that's hugely enabling for lots of diagnostic technologies. And of course, there's been lots of advances in sensors, an area that is also very important to do diagnostics. But despite all of this, there's been some there continue to be significant gaps. In fact, in some sense, the COVID honeymoon is over for diagnostic companies, and you know, being relevant in the post-COVID world is very important. Actually, two of the companies that I listed in that page, when I made that slide, it was about a year ago. In the meantime, two of those three companies have declared bankruptcy. So it was a huge rise and an equally sharp fall. So I think you need a story, obviously, beyond COVID, and you know, this is a process and evolution for everybody. The second, I think, many important learnings have come through COVID. One is it's not enough to just give a test result. You need to close the loop, especially if you are in a POC setting, being able to say what to do next, especially if you're going more and more towards consumers, giving them further information beyond a test result is important. The other interesting one is, this is reported by many people, and all of us have seen it. You feel you have some flu-like symptoms, good chance it's COVID, you test COVID rapid, and you come negative, right? But then the obvious question is, then what? You know you're sick, you know you have something, but all you get at that point is, I don't have COVID. So this is very unsatisfactory for most people. They want to know, I have something. It's probably flu, maybe it's something else. So being able to do multiple tests and ruling in as opposed to just ruling out is also very important. Another kind of important thing which came up again is lots of packaging, etc. These are not areas that are thought about by typical diagnostic companies, right? Because you're selling to trained users. The moment you go closer to home, learning from the big electronics companies, the consumer electronics companies, in terms of, Apple, for example, being a leader in this, very simple instructions, just saying do this, and in big font, maybe small things like that are very important in order to enable home testing. There's also a lot of solutions around. Most of these are lateral flow based solutions, which are nitrocellulose membranes coated with antigen antibody, and they come with a portable reader. So despite the existence of these, none of them, I would say, have made a massive impact in changing the way diagnostics is done. So all of these are some of the gaps that we are focusing on as a company in terms of solving. So I'll just switch gears a little bit, go back in time. This is some work from my PhD thesis, and I'm trying to connect some of that to the work that we are doing today. So this is the primary work for my PhD thesis was actually coming up with a technique to make colloidal hydrogel sensor particles. Actually slightly larger than colloidal, like tens to hundreds of microns. But the basic idea here was that you actually shine UV light into a photopolymer, which is sandwiched inside PDMS, PDMS being a siloxane rubber material, which is actually very porous. So what we found, interestingly, is that when you photopolymerize something, you form an object, but it doesn't stick to the bottom and the top, which allows you to flow it out. Most of the time when you do photolithography, if any of you are familiar, you are photocuring an object on a surface and it sticks to it. So this was one of those accidental discoveries which turned out to be quite important. And that was because of oxygen, which diffuses in through the PDMS and allows inhibition reaction to happen at the surface. So using that, this is the flow lithography technique we developed. So you can actually photocure and flow out colloidal particles and make them in very large numbers. So this technique is called flow lithography, as opposed to just say lithography on a stationary material. And with that, you can make large numbers of these colloidal particles. And these colloidal particles are interesting because they are unlike standard photochemistries, which are quite toxic and not very biocompatible. You can make these in hydrogel materials. You can put oligos, you can put antibodies, enzymes. You can really functionalize these beads. And you can even control their porosity. You can control lots of their attributes, which make it important for use as sensors. So now just coming back again to our product, this is what our consumable looks like. And at the core of it, in some sense, are these hydrogel-based beads, which we use for our multiplexed analysis. But some of the important features of the platform that we've built are the following. One is you can do multiple analyte detection. The second is we're able to integrate in this consumable both dry and liquid reagents, so you don't have to do any manual work. We're also able to integrate key features like capillary blood-based plasma separation. And of course, the whole thing is very portable and low cost. And this works together with an instrument, which is a small portable device that has also got connectivity features. Because the idea is you want to do this in a doctor's office, you want to do this maybe in a pharmacy, you want to be able to then do some active troubleshooting, remote maintenance, etc. So this can actually talk to the cloud. You simply change cartridges to do different kinds of assays. You don't have to change the instrument. And it's an image processing based technique. So we have a camera that runs some machine learning algorithms that can do the test. So the core idea here in some sense is that we have these shape encoded hydrogel beads. For example, this is a thyroid panel. So doing TSH, T4 and T3 together. In some sense, of course, these are very well-known tests. But what we're trying to do here is do them from a capillary blood sample, which means use only 30 microliters of blood, whereas today, of course, you would need few hundred microliters of blood to do it. So it's technically challenging to do it with a very small volume of sample. That's where the microfluidics gives you some advantage. And we have shape encoded beads like this. So the TSH is, let's say, a hexagon, the T4 is a rectangle, and the T3 is a circle. So we use shape encoded beads, and we do a final fluorescence readout of these shapes and correlate and get these values from them. So this is maybe just kind of showing you a video of how the whole platform looks. So that's the instrument, as I just said. And this is the consumable. And the entire thing works directly just by application of the figure stick. So all of the other reagents are actually loaded inside here. So you take capillary blood, of course, and you wipe off the first, and then you apply the second drop directly to the cartridge. The user knows that there's enough blood because it lights up there to be read. And at that point, you can load the cartridge into the instrument. You don't have to do any other manual steps at this point. So the first step is actually a metering step. So let's say you add 35 or 40, you only want to take 30, correct? Its volume is very critical here. So we've got a metering chamber which will only pull out 30 and then discard the rest. Then it's mixed with some buffers and fluorescence reagents and so on. There's a mixing incubation steps that happens inside the instrument. And as I said before, these hydrogel beads which are inside the cartridge actually glow fluorescently at the end of the assay, and their fluorescence is proportional to the concentration of analytes. And each of the tests is shape encoded, so you can use image processing to back calculate which test gave what. And there is a fluorescence based system that's inside the instrument that's able to read those cartridges. So that's the idea of the test, just to give you a sense of how the internals of it work. So you, of course, dispose that plastic consumable, and your test results show up on the screen like this. Okay. And we've done quite a bit of clinical validation of this platform, which is published as well. So these are, in fact, some fertility hormones where we've compared chemiluminescence, which is the gold standard technology, with the values from these hydrogel sensors, got good data to show that the platform is capable of giving good quantitative data. And I think in terms of what is the key value proposition of our technology compared to the competition, which is really portable LFA-based tests, one is good quantitative performance. I think good quantitative performance seems easy to say, but it's actually quite challenging to get a good repeatable performance with low precisions that is under 10 or 15% CVs on existing technology. So that's something that we're able to do because of the microfluidic technologies and the bead-based chemistries that we're able to control. We're able to do multiplexed panels, so we can do, let's say, an entire tropical fever panel or a respiratory panel or a hormone panel with only 30 microliters of capillary blood, which is something that's not possible to do with current technologies. And finally, it's load and go, which really means from a user point of view, you only have to add sample and you don't really have to do any other workflow. As part of doing this, one of the things that's happened as a company is we've built an entire toolbox of microfluidic technologies, whether it is storage chambers for swabs, metering, plasma separation, mixing. These are all things that you take for granted that you might just do in the lab in an Eppendorf tube. But the moment you want to automate it at a small volume without, let's say, a fancy liquid handling system, these can be surprisingly challenging to do. So we have an entire toolbox of these things built out that can do assays. And what we are looking for, since I think part of the idea of being here at CDRI is, working with maybe some of you people tomorrow, is we're looking to build more and more assays on our platform. So if there are diagnostic assays that you people feel are relevant, that you think are important clinically, commercially, we are happy to talk to you people, work with you on building assay automation in these sorts of miniature portable formats, and of course all of the instrumentation that goes with it. So we are working on several tests right now. In fact, because of the partnership with CIPLA, there's many POC tests, and I'll just point out a couple of them which are quite interesting. I think one is, of course, a tropical fever panel combining dengue, malaria, chikungunya, lepto, typhoid. Similarly, a respiratory panel combining flu, RSV, COVID. So these are the kind of things which I talked about earlier, which will help you hopefully rule in and not just rule out when you have certain symptoms. So these are things that we are working on at this point. We also have a manufacturing facility to make these cartridges at scale. And this is something that's also, again, an important capability to build. These are not things that are available in India today. So being able to make these microfluidic cartridges at scale is something that is also a non-trivial task, and something that we are able to do under a certification from various government authorities. Just to kind of maybe talk a little bit about the molecular side, I know there's some interest in HPV and cervical cancer. So working in collaboration with some others, including GenePath and Firebird, we've actually built a multiplex molecular assay. So this is for HPV, where you can screen, up to 11 different genes using one sample. So starting with the swab, I think those who know HPV well know the HPV 16 and 18 are the ones that you need to be concerned about, but there's several other HPVs which are less important or kind of more benign. But with this test, we're able to differentiate the specific HPVs and also look at all of the other HPVs down to about 20 copies per microliter. Starting with the same sample, we're able to do a highly multiplexed molecular reaction. So we've got our platform is tuned to be able to do these kinds of highly multiplexed molecular assays as well. And this is at an earlier stage of development, but looks something like this, where you have again, plastic consumable combined with this instrument that enables you to do this testing. I just kind of briefly maybe close with a couple of other things, because I remember you had specifically discussed, he asked about fabric-based diagnostics. This is something that we spent quite a bit of effort on, and actually built a fabric-based platform to compete effectively with LFA. So LFA is, of course, a nitrocellulose membrane. In this case, what we do is we have woven fabric. So you can see this is a woven piece of cloth, and we coat thread or yarn with different reagents, and then we weave them in here. So you might have a test line, you might have a control line here, and then of course you have your absorbing material. And simply by changing the kind of weave that you have, you can change the kind of pattern of fabric. So you have a highly absorbing zone here, you might have different kinds of flow properties here. So we've built, I would say, the world's first effectively woven fabric-based sensor platform. And then you cut these into individual strips. So we've made both lateral flow immunoassay kind of tests, as well as electrochemistry-based tests. So here we've taken electrodes, which are basically coated with carbon ink, and then woven them in. And we're actually able to also have a silk is interesting material. You can make it both hydrophilic, hydrophobic, depending on the treatment you give it. So you can actually see this little white zone here. It is very hydrophilic. So when you apply blood, it will only wick here. It will not wick on the rest of the background. So you can do some very interesting things with this kind of thing. You can also do some electrophoretic. We've done isotachophoresis, electrophoresis on this. So we've kind of really built out this fabric-based platform. Today, there's lots of interest in wearable technologies to do testing, let's say, directly from sweat and other body fluids. So this is the kind of place we believe it could be quite interesting to apply it. Finally, the other product that we have is an IUI-based a kit to do intrauterine insemination, rather to select sperm for your intrauterine insemination, which is a low-cost technique in the IVF world compared to, let's say, ICSI. But the problem is that the number of pregnancies is only about 10 to 15%. So you have very low percentage of pregnancies that happen in IUI, where many gynects would like to do IUI, provided they could get increased percentage of pregnancies. And today, what happens is most of the sperm is chosen based on motility. So density gradient or swim-up techniques use motility as a way to separate sperm. However, it's been demonstrated that HA, which is a molecule that's actually present on the oocyte in the natural reproductive system, is actually a good selecting molecule for mature sperm with good DNA integrity. So that's what we do. We actually use HA binding. So we have magnetic beads with HA on them, which are then pulling out sperm. And then that sperm is released with an enzyme and is actually used for IUI. So this is a product that's quite simple in concept. You have magnetic beads that bind to HA, use a magnetic stand to pull them off, and then you release it, and then you use that prepared sample for the IUI. We've done almost 5 years of work on this, including one clinical trial, which was with ethics, consent, and approval, and so on, where we were able to demonstrate a significantly improved pregnancy rate up to about 20 to 25%, done at Manipal Fertility Center in Bangalore. And this is something that's quite interesting because it's a very simple way to increase pregnancy outcomes for IUI. So this is a product that is also got a US patent and is commercialized right now by Bharat Serums and Vaccines across multiple centers in India. So this is the other product that we have, which is not really a testing product, but more of a preparation kit, you can say. I will finish up by just acknowledging some of our partners and collaborators, and thank you all for the chance. Sure. Yes. Yes.
How long the device will take to install?
The total time to result is 25 minutes.
25 minutes.
Yeah.
So that one sample will run doing all the process.
Correct. One sample will do all 3 in 25 minutes.
And when you correlate with the other wet lab results, it's like equally similar? Or
Yeah, we've got, I mean, the R square across, let's say, 100 samples is greater than 0.9. And so you have good accuracy. I would say some of the lower limits of detection, we are maybe not as good as the best high-end lab techniques, but better than LFA.
And whatever it is, you have showed all blood-based?
Yes, it's all blood-based. Actually, it's not serum-based. So that's the other important thing, that you can do directly from a blood sample as opposed to serum.
I mean, in fact, I was just raising the point in the evening whether urine can be tried for such tests.
Yeah, I think it's interesting. Urine, even people are trying saliva now. I don't know whether urine, things pass at a much lower concentration in urine. Saliva might be a better non-invasive method. In fact, cortisol, for example, people do today with saliva. And there are many other things people are looking at doing using saliva today. It could work on either, but the whole idea is that venous blood is more challenging to get in a POC setting. So it should be able to work with capillary volumes.
Besides the TT report, you said you are doing other tests also.
Correct.
So they should be pitched twice.
No, that's a good question. So ideally, we are focusing on scenarios, and that's where the multiplexing becomes important, to make a minimal set of tests which are relevant clinically.
That's for me as a pathologist.
Yes.
Labs, private labs,
Yeah.
will be a better target for you than the
Doctors. No, there is a fair point there. I think in fact, the small labs are the more ideal customers. But the push to the doctor's office is also happening for another reason, that
I know.
from a commercial point of view,
Economic, but the thing is that they
yeah. But see, once you go to a lab, there is probably a phlebotomist as well. Yeah. Yeah. So that's where I think selecting the panels is important. For example, if you have a fever panel, in that particular clinical context, you might only be looking to do that.
will be doing.
Correct.
So venous blood will always be there and stored like that. So focus. Is there any difference between capillary blood and venous blood? That will be.
Yeah, so this is a subject that others have studied as well, capillary versus venous. For example, glucose, it is very well studied, that provided the sample is collected properly, this is the big thing, and I think Radha also knows this well. Provided the sample is collected well, the correlation is good. But if you don't collect the sample well, there can be significant errors. Yes. Yes, as of now, we have more focused on these immune markers, but we have in early stage something which closer to a lipid profile, you know, and things which are related to diabetes as well. So those are all more chemistry tests, clinical chemistry based tests. Most of the ones that I showed use antibody, antigen, immunoassay type thing. Correct, and HbA1c as well, absolutely. So lipid profile plus HbA1c is probably the most relevant in that context which you have mentioned.
Great.
Yes.
Are you considering, like, with your background, microfluidics background, for CTC detection or from the blood patients?
Yeah. See, I think with CTC, by the way, unless you start with a significant volume of blood to begin with, it is difficult to get because you might have, let's say, 5 cells per ml or something like that. So there's no advantage to starting with a smaller volume. In fact, it's a problem if you start with too small a volume, right? Secondly, I mean, those are probably better off done in a tertiary setting than in a POCT setting, because it's a very important test. So you can use a microfluidic engineering for sure, but I think it doesn't necessarily benefit from a small portable form factor or low sample volumes.
And also, how the people, like patient, will respond to immunotherapy, like PD-1, PD-L1, like that kind of detection in from whole blood tumor cells or something.
I think anything which is repeat testing, even for standard, let's say, kidney function tests and so on, these are also good examples of things for POCT where you might want to visit a doctor every few months or at home even do repeat testing if you're on dialysis, for example. These are good markers to look at.
Very great talk. So one question I have is that because you the T3 TSH you based on shapes, right?
Yes.
So what happens if there is a change in the shape?
No, we are designing those shapes. So we are not they won't change without us changing them.
Okay, so you can do the multiplexing in that sense.
Yes.
So probably
Correct. So if we had a new test tomorrow, we would just introduce another shape.
Okay. So next question is that how about you think about the variables as the biosensors? Can you, because you showed some of the pictures where you showed that the silk could be used as a material, right?
Correct.
So you think of envisioning variables as a biosensors for detecting diseases, like example?
Yeah.
Like,
no, it's an area in itself. Honestly, you almost have to start another company to go do this. I just showed it because it's a technology that we've developed. There is a huge interest right now because, of course, most of the variables today are focused on electrophysiological measurements like ECG, EEG, those kind of things. Less on blood analyte or fluid analyte testing, except for CGM, continuous glucose monitoring, which has become a big movement right now. So I think if you kind of look ahead, there will be a lot more of those kind of things, including for, say, drug monitoring trials and so on. Many people are interested in, let's say, continuous monitoring of toxicity profiles. So forget even home use and so on, but in drug trials, this can have a massive use because you will get continuous data as opposed to, let's say every day or a few hours. So I think there's a lot of scope for wearables for the right application.
Okay, because one more is that because when you showed that the electrodes are also could be used.
Yes.
So can you design some sort of ECG patterns to detect the ECG patterns for the?
You could. I'm pretty sure you can. There are solutions out there which do this quite well as well. There are companies that provide, even single lead and 2 lead, 3 lead ECGs. There are solutions out there today for that. Yes.
I also have a question. Can microfluidics be used to detect number of DNA or RNA molecules in a sample also? Because you're detecting protein samples by conjugating them to antibodies.
So I showed the HPV test, for example. That's a DNA test, right? You are doing some amplification, and there is a lamp-based chemistry in that case, which is doing the final readout. So you certainly can.
So gene expression levels can be measured using microfluidics?
Yeah. See, microfluidics is just the engineering toolbox, right? What it's enabling in this context is with as little as, let's say, 3 or 4 microliters of elute, I was able to run, which you might not do in a standard, let's say, PCR tube format. And you can even go further down, let's say, to half a microliter, 1 microliter. But the chemistry is no different than like attackman chemistry or lamp chemistry that you might be using.
Right. So if you isolate RNA from a patient, convert it into cDNA, and load 10 microliters into it,
yeah,
it can detect how many
exactly
molecules.
See, but the advantage of microfluidics is all of those steps that you just mentioned should be automated,
Right.
because you would like to just say, put swab into this cartridge and load the cassette in this POCT context, because you don't want to do any extra step, right? That is the kind of things that can be enabled with these technologies.
Thank you.
Yes. Yes. Yeah, antigen antibody based, and if you know this area, many of them are actually only looking for antibodies. For example, if you look at typhoid, there is no good antigen available that is to be detected. So the typhoid tests are still basically looking at IgG, IgM.
So what can you comment on the MOIs? How many MOIs it can detect? What is the minimum inoculum it can detect, or based on that? Because sometimes, different patients would have different amount of the response.
Yeah.
So what is the minimum limit of detection? It would be definitely different for different pathogens.
Correct.
So how do you make it in a readable format that, for all the 3 pathogens or whatever multiplexing you're doing?
Yes.
So what would be your limit of detection, that this is the limit of detection for these different pathogens?
First of all, this assay is reasonably complex, because let's say you have a bunch of antigen assays like malaria, PFHRP2, PVLDH, etc. You don't want to dilute very much to get good sensitivity. Whereas let's say an IgG assay has to be by definition diluted to avoid all these hook effects and so on. So what I've glossed over a little bit is there are about 2 or 3 different paths actually for the multiple assays to happen and different workflows. So this is exactly the kind of thing that's difficult to do today with LFA. You have to take so many strips to get all of this data, correct? That's number one. To your second question, how do you correlate IgG, IgM levels to inoculum? Even I'm not aware actually, because you're not even directly measuring antigen, right? Which at least in malaria, you can correlate, say, PFHRP2 antigen levels to malaria number of cells and so on. In typhoid, I'm not sure even if what is the answer to that question actually.
Okay. Thank you.
Yes. Yes.
You're doing one sample at a time.
Correct.
We have seen cassette. So is it possible to miniaturize it further and have multiple samples?
Yeah, it's quite small, and I think one of the things, by the way, you run into commercially is if you make things too small, people feel it's probably not very valuable. So it has to have certain heft. I think it's true, but there's jokes apart. There's no advantage to miniaturizing beyond a point unless it's truly for field testing, which has to be carried in your hand or your pocket and like our phones taken somewhere, then yes. But I think this is a lab instrument. I mean, this is something you can hold on the palm of your hand. So while we are, of course, always trying to miniaturize, I think for this configuration, we're good. But to your other point, that can you not do more samples at a time? Yes. There also in POCT, the way people have thought about it is modularization, right? That one instrument is basically 1 into n, and you do 4 or 8 or something, rather than the standard lab automation format where something is moving around. Yeah. Yeah. So I think one misnomer generally is that POCT should be cheaper than lab testing. In fact, if anything, it's probably more expensive because you're amortizing, let's say, a consumable which has got some cost associated with it. So I think the cost advantage plays when you can do more multiplexing. Effectively, all of the engineering that goes into making quite a complex cartridge, if you can amortize over 5, 10, 15 tests, the economics start becoming better and better. But if you're doing only one test, then the LFA or an older technology is definitely going to be cheaper. And you can't beat the lab consumable chemistry, because effectively, let's say what is Siemens selling to Thyrocare? They're just selling antibody solution almost, right? That's their only variable cost. There's no other cost really, and at tremendous scale. So if you try and compete with that unit economics, it's impossible. And I think that's why this need is finding tests which are really needed at the point of care is important. Yes. Yeah, no, that's a good question. I think, and a big challenge as well. Uh, Providing quantitative stability, right? Not just stability, but quantitative stability is actually the significant challenge. In fact, one of the things that I would be interested to work on, and I was asking your NMR and other teams about it, is how to study quantitative stability, maybe with some of the equipment that is here. In our case, we're using hydrogel-based beads. They themselves provide some protection to these reagents. And currently, our storage conditions are at 4 degrees. It's not room temperature. So at 4 degrees, let's say a year. But then, sorry? 1 year. Yeah, with some maths and various algorithms associated with it. But ideally, people are looking for 2 years room temperature. That is what the need in the marketplace is, which is actually a very significant burden. So I think finding ways to do that is a big challenge and something worth working on. Yeah. Sure, we should have a conversation. Okay. I think if there's nothing else, thank you all for the opportunity.
Thank you, sir, for sharing with us very interesting multiple innovations. Now it's time to felicitate our guests. Please join me in inviting Director Ma'am to honor Dr. Manjari and Dr. Dhananjay. May I please request Dr. Atul Goyal and Dr. Ashish Arora to join? So let's felicitate Dr. Manjari. Thank you, sir. Thank you, ma'am, for very inspiring talks. Now, I request Coordinator of Outreach Program at CDRI, Dr. Sanjeev Yadav, to propose vote of thanks.
Good afternoon, everyone. I feel honored and privileged to get the opportunity to propose a vote of thanks on this special occasion. On behalf of the organizing committee and on my personal behalf, I would like to extend my heartfelt gratitude to the esteemed guest, Dr. Manjari Bakre, CEO Onco STEM Diagnostics Private Limited, and Dr. Dhananjay Dendukuri, Chief Executive Officer and co-founder of Achira Labs, for sparing their valuable time for delivering such a wonderful talk. I wish to thank our director, for her continuous guidance and support for organizing this event. And I would like to thank all esteemed guests who have joined us today and blessed us with their presence, particularly Dr. Kamboj, who has joined us today. I wish to thank Dr. Atul Goyal and Dr. Ashish Arora for their initiative and their efforts for organizing this event. And I am also thankful to all annual report editorial board members, particularly Dr. Anand, Dr. Shruti and Bhavna, for their efforts for timely release of annual report. And I would like to thank photography and auditorium team for their efforts to organize this event. I would like to thank all the audience present in the auditorium for their presence. And in last, I would like to thank all the press and media people for spreading the words through their potential media. Thank you very much. Now I request all to please stand up for the national anthem, please.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे